The protein science facility, 90% of our clients are asking us to do mass spectrometry-based analysis. However, a small percentage of our clients would require us to help them with protein purification and isolations. Some laboratories are not equipped to handle large-scale isolations and purifications. So we'll help them with different chromatography steps and purification steps. So we are set up with different HPLC and FPLC instruments to help them with those analyses. We also sometimes help them with some analysis such as gel electrophoresis, one-dimensional and two-dimensional gel electrophoresis. We also help with image analysis from gel electrophoresis. There is also a small component of the protein science that we actually study protein-protein interaction using an instrument called beer core. This beer core uses the principle of surface plasmon resonance, SPR, in analyzing protein-protein interactions. So SPR is something that we also do for protein interactions. Most of our clients are from the university community. We have a client base of approximately 150 groups of researchers. So they come from engineering, chemistry, biology, physics, crop sciences, and the vet school. There are basically 4 different things that we offer to our clients. The first and the most sought after technique that we do is called protein identification. Chemists and biologists, they often work on fractions and samples, but not knowing the different components they have inside their sample. So using mass spectrometry, we can identify the different protein components and tell them what players they have present in their sample. So that's the first one, protein identification. The second one is closely related to the protein identification, and that is to quantify the different amount of the different protein components. Let's say you have 3 components in your sample. And maybe depending on the expression of the different components, call them A, B, and C's. If level A is high, maybe that would affect the amount of B and affect the amount of C. So understanding the amount of A and B and C in the sample is very, very important. So that's called quantitation. The third one is a little bit more complicated. Since proteins can be modified in many, many different ways called post-translational modifications. So these are chemical modifications that modify the chemical characteristics of the proteins and causing them to fold differently and to function differently. So in other words, the protein would be changing their characters by having the different post-translational modifications. When proteins are modified, how are they different? Typically, proteins would have different partners, just like humans. We have friends. We always gang up with our good friends. And each of those protein components, they usually work in gangs, except we don't call them gangs. We call them interactome because they interact together, or you can call them proteome. So interactome and proteome are like a group of proteins that they associate with each other. Let's say a protein normally, when it's not modified, then it would have an interactome of a certain different components. And then after that protein is modified with post-translational modifications, it would pick up new friends. It will pick up friends that we never would have expected. And then the synergistic effect of that proteome would change, and then the function of that protein would collectively change and serve different purpose. So that is very, very important. And the fourth component that we do for our client is to take all the information that we have helped them analyze to, first, the protein identification, the quantitation, the interactome, all link all the information together in a bioinformatic picture. And so to give them a sense what really is inside the biological system, inside the cell.